Welcome back. In the last class, we discussed something about multinuclear NMR and some isotopic shifts, effects of substitution of different isotopes on the NMR spectrum. I discussed quite a bit. And I showed you how important it is just by mere substitution of replacement of one proton by deuterium had tremendous effect on the spectrum. It moved it quite a bit. Similarly, especially I saw and when we discussed halogen substitute like fluorine, chlorine, bromine had a tremendous effect in giving rise to isotopic shifts. And I showed a couple of examples like difluorobrom, I think fluorodibromo or so one molecule or CFCl3, PCl3, PTL6, so many molecules were there. And we showed the example of couple of them and we worked out the intensity pattern how it comes based on the population distribution of different isotopes. So this is a very important point. Please remember, we need to understand. And then in the last slide, I did not have time. I was in a hurry. So let me go slowly in taking 1 or 2 minutes today. This is the spectrum of deuterated analogues of vinyl fluoride. Remember, I took the SNH3 minus spectrum, tin 119 NMR showed, where I took deuterated analogues also there itself. Then we could interpret this NMR spectrum of that so easily, taking into account what happens if one deuterium substituted, two substituted, all the three deuterium. We could explain the multiplicity pattern. Now, this is a similar molecule. There is a 119 tin NMR. Now it is a 19F NMR, and this is the molecule. Remember, it is a proton decoupled. Right? I am looking at fluorine NMR with proton decoupling. So completely all proton couplings are removed. So if this is the molecule, in the only one fluorine is substituted, when no other couplings are there, of course don't worry about carbon 13 satellites, we don't worry about it. When all the protons are completely decoupled, how many peaks we expect? Only one peak, exactly what you see. Now, the deuterated analogues of this was taken. In the sense, di-deuter analog will take first go to mono and then go to di. If one of the deuterium protons is replaced by deuterium, one of the protons, how the spectrum affects you will see. First one, let us consider this molecule. Very interesting. You see here what is happening. Now, as far as the fluorine is concerned, it is 3 bonds away. 3J FD coupling is there, but it is cis 3 bond vicinal coupling. Vicinal coupling, it is a cis coupling. It is a cis orientation. And the coupling you can easily measure. This deuterium, because it is spin 1, splits this fluorine resonance of this one into 3 lanes of equal intensity. And you measure the separation of adjacent peaks of this triplet, you are going to get F1 decoupling corresponding to vicinal cis configuration, 3J coupling, 3J F decoupling. Okay, that is fine. Next, we will go to other one. Now, this deuterium is in transposition, not in cis position. Again 3 bond coupling, but it is moved from this position to this position. Look at the pattern. What does it tell you? The J coupling is much more now. Trans coupling is much more J coupling. See, quite large. So we are going to see FD coupling quite larger like this. What is this one? This is a situation where deuterium is geminal to fluorine. In the sense, it is only 2 bonds away. 2 bond FD coupling is much larger than 3 bond coupling. See the separation here? Quite large. Now measure this peak separation between this and this, you get JFD, 2 bond JFD. Like here, you measure this separation to this separation, you get 3 bond FD. Here you are getting 2 bond fluorine type deuterium coupling. Very easy, right? So that's what you are going to observe when you have a single deuterium with a different analogues where deuterium substitution position is varied. Now look at this molecule. Now we are looking at two deuterium substitution. Deuterium is substituted at two positions, one here, one here. Interestingly, what happens is one of the deuterium splits fluorine into three lanes of equal intensity like here. But what does the other deuterium does? Remember, they are non-equivalent. You cannot use 2Ni plus 1 for 2Ni plus 1 formula here because they are non-equivalent. First, one of them will split into three-line pattern of equal intensity. Second deuterium splits each line of this three line into three lines of equal intensity. You understand? So it is a deuterium fluorine coupling of, because of spin 1, deuterium gives rise to three peaks, and each line of this three lines further split into three lines because of coupling with another deuterium. So you see, you are seeing 9-line pattern here. 9 lines are there. Exactly same pattern you will see in this position when the deuterium is in the 2 bonds and fluorine is experiencing 2-bond deuterium coupling and 3-bond deuterium coupling. See, this is what it is. One of them is a 2-bond coupling, other is a 3-bond coupling. How do you identify 2-bond coupling? Simply take the center of this to center of this, or center of this to center of this, gives you, it should exactly match with this. It's a 2-bond coupling. And then each line of this three line is further split by another deuterium into three lines. Very clearly 9 lines you can see. That is the beauty of coupling with the second deuterium, right? Now this is the another analog of that. Another very interesting thing. Now we have a geminal coupling, I'm sorry, I would say 2-bond coupling of fluorine and deuterium, and 3-bond coupling to deuterium, which is in trans position. And you see again 9-line pattern. Analysis of that gives me 2-bond FD coupling and 3-bond FD coupling trans to fluorine. Very nice, you see. And most interesting, you have to understand how the deuterium substitution has a dramatic effect on the chemical sheet position because of isotopic substitution in the position. Secondly, here two deuteriums are substituted. Here is no substitution. In one substitution, in a different position, it shifted like this. With two deuteriums substituted, even more it shifted. So the dramatic effect of the substitution of deuterium is seen on the chemical shifts of fluorine also. Okay, with this, now we'll try to analyze some of the spectra of different nuclei. I wanted to make you understand heteronuclear NMR. Extensively, I have covered carbon, proton, everything. That is more than sufficient for you because with that knowledge you can analyze. But some tricky situations will be there, like we saw isotope shift or varieties of other things will come. Some of the problems once in a while we come across. So let us see some of the spectra other than proton and carbon, some heteronuclear spectra. Heteronuclear in NMR jargon is anything other than proton. Okay, that's called heteronuclear. Of course, heteronuclear means anything. But when carbon is observed, proton is heteronuclear, doesn't matter. But NMR colloquially we say, I'm looking at heteronuclear spectrum means other than proton. This is a convention. Of course, it's not that you should not see proton. Okay, now let us look at the fluorine spectrum of parafluorophenol. Phenol is C6H5OH, and para position in the 4th position, we are having fluorine. If you see the fluorine spectrum of it, fantastic. It's beautiful line you are going to see. When the same thing is expanded, it is a multiplet. You see why this multiplet is coming? See, first thing, fluorine is experiencing coupling with these two protons which are ortho to it. They have same coupling strength because of symmetry, right? There is a C2 symmetry for the molecule, same coupling strength. Then it gives us a triplet like pattern. because two peaks of the center overlap. Each of these things experience coupling with metaprotons of equal strength. Both metaprotons have same coupling strength to fluorine. Now each of this triplet line pattern further split into triplet because of that. So you should get triplet of triplets. So you should get in principle 9 lines. We will try to understand 1, 2, 1, 1 line, 1 triplet, 1, 2, 1, 1 triplet, 1, 2, 1, 1 triplet. So all the 3 triplets are here overlapped. And this is the fluorine spectrum. If you analyze this, you can get which is the larger triplet, which is the smaller triplet. If you analyze, you can get coupling of fluorine to orthoprotons and also to metaprotons. Very easy. Okay. This is a simple fluorine spectrum. Now I look for the phosphorus 31. Phosphorus 31 is, this is spin half nuclei and it is another favorable nuclei. Abundance is 100%. Easy to detect. As I told you earlier, spin off nucleus are really friendly for us. Very easy to deal with them. Unlike quadrupolar nuclei with spin greater than one half, like 1, 3 by 2, 5 by 2, 7 by 2, as the spin keeps increasing, it is very, very difficult for us to handle. So it is more comfortable to work with the spin off nuclei and also with 100% abundance. So that is one such nucleus, P31. Now let us look at the P31 spectrum of this molecule. Okay. What type of spectrum we expect here? Look at the spectrum. We will try to understand. First thing, all the 3 fluorines are equivalent. Okay, all the 3 fluorines are equivalent. That will split phosphorus into a quadrat. Remember, it is split into a quadrat. And then this phosphorus is also split by proton. What will happen when it splits? It splits each NH quadrat into a doublet. You understand? Now, the question is, which is larger? This FH coupling is larger or HP coupling is larger? Accordingly, you will say it is a doublet of quadrat or quartets of doublets. See, you understand now. Look at this, which is larger here? See, this is one doublet of quartet is 1, 3, 3, 1. This is 2 lines, 1, 3, 3, 1. It is a quadrat. Each line is a quadrat is doublet like this because of coupling with HB. Now what you have to see is which coupling is larger. Measure this coupling. This is a quadrat. Okay. Or measure this to this one. Which of them is larger? If fluorine to phosphorus is larger, that coupling is larger, then quadrat coupling is larger. Doublet is smaller than that. Okay, in principle, take any of the P, measure with respect to this. Yeah, I can consider, for example, I will take a highlighter and I'll show you this peak, this peak, this peak, and this peak forms one quadrat. This, and this form another quadrat. So now this quadrat, if you take 1, 2, 3, and 4, find out the center of that. Let us say this is the center, and find out this quadrat, and find out the center of that. This is HP coupling, and this is PF coupling. Which is larger, we find out, because it looks like more or less they are coming equal. Depending upon whichever is larger, you call it quartet of a doublet, or doublet of quadrat. Okay, that's what you should understand. Okay, this is one quadrat, this is another quadrat. This is JPF, this is JHP. Looks like that JPF is larger than JHP. As a consequence, it is a quartet of doublets. Now look at the other phosphorus spectrum of another molecule, which is proton decoupled 31p spectrum. How many protons, how many phosphorus is there? Phosphorus spectrum, if you are looking at, this is one phosphorus, This is another phosphorus, and this is, three phosphorus are there. Okay, and all the three phosphorus are chemically inequivalent because there is, see, if you look at the structure of the molecule, there is no symmetry. So all are chemically inequivalent. There must be three phosphorus peaks we get. Okay, one is here, Px, one phosphorus. There are two phosphorus here, which are overlapped. Both chemical shifts are there, Pa and Pb. Very small change in the chemical shift, they are overlapped here. And you are not able to see that at all. You cannot identify what is what. But if you carefully see, you are going to see some peaks here. You are going to see some peaks here. What are they? Okay, these are all coupling of phosphorus with platinum. It couples to platinum. Platinum abundance is small. Then what do you get? Satellites. You are going to get satellites. That's what happens. Now this you are going to get satellites for this peak here. This is phosphorus platinum coupling. For this Pa and Pb, you get two satellites. Okay? How can we get two satellites? That means you have two phosphorus chemical shifts more or less overlapped. But interestingly, its coupling to platinum are different. One coupling may be this, other coupling may be this. Now, to identify which is the chemical shift of phosphorus A and which is the chemical shift of phosphorus B, simply take this separation, divide by 2. That is chemical shift of one phosphorus. Take this separation, okay, and divide by 2. That is chemical shift of other phosphorus. So, what you cannot distinguish chemical shifts very clearly in the phosphorus spectrum, you can make use of satellites, and because satellite has better resolution here, you can make use of the satellite resolution and make the assignment of the individual phosphorus. So very beautifully, of course, coupling patterns can be understood easily. Okay, now we'll go to boron NMR. Boron is something which, borosilicate classes are there. NMR tubes sometimes contain boron. They made up a borosilicate class. So if you are looking for boron, you should not use the borosilicate class because you get boron signal from the NMR sample tube. So advised thing for such situation is to use quartz tubes. Remember, you have to use quartz NMR tubes when you want to study boron NMR. One interesting thing is boron spin is 3. Spin is 3 for boron 10. That's very interesting. So if you want to see the boron NMR of, let's say, BH4, you get a single peak because we are looking for simple, this of course okay, this is not BH4, it is BF3 ETO2. Okay, so you are going to get only one boron signal. Of course, you should couple to fluorine. Let's see now what is happening in the next one. In the next one, if this is BH4, that is BF3 ETO2 as the reference. Now in the B10 NMR, if I see B10, this H4 should couple to that. Am I right? Go back here, remove everything, look it. Coupling to proton is not usually observed here in the smallest and most symmetrical molecule except in these molecules. So you don't see coupling to protons. But look at here. Boron 10 NMR spectrum of this BH4, H4 is 4 chemically equivalent protons. What you have to do? You have to apply 2 Ni plus 1 rule. When you apply 2 Ni plus 1 rule, follow Pascal triangle because proton is spin half. Boron, when you are observing, you have to see the coupling with proton. But in this example, what we are seeing is 5-line pattern where it is coupled to boron 10. It is a boron 10 NMR. There is also boron 11. We'll come to that later, how that spectrum comes. The boron 10 NMR, boron is coupled to 4 protons. Using the Pascal triangle, you get 14641 intensity. This Peaks adjacent separation gives you boron 10 and proton coupling. So B10 H coupling. It's a proton coupled NMR spectrum. So why you got 5 lines? H4, 2Na plus 1, you got 5 lines. Intensity is Pascal triangle for you apply, and adjacent peak separation gives you J coupling. So boron 10 NMR spectrum of BH4 is easy to interpret. And also it is done in D2O. No question of solvent peak or anything here. Very simple. Same thing to boron 11 NMR. What happens now? Pattern remains same. It cannot change. Because again, doesn't matter what is the spin of the observing nuclei. This spin is different. Whereas this spin is different. Boron 10. This is spin 3. This is spin 3 by 2, I think. Boron 11. Doesn't matter what is the spin of the observing nuclei. But what it is coupled to, that is proton. So again, 4 equivalent protons. Apply Pascal triangle, 2Na plus 1 rule. Get the intensity 14641. Exactly same pattern what you saw here. So you get B10 proton coupling. Here you get B11 proton coupling. See, isotope couplings are different. B11 proton is 65 Hertz approximately. This is 27.3 Hertz. See the difference? B10 proton coupling is much, almost half of it compared to 11B coupling. So very easily we can see boron NMR and its coupling to proton. Especially in this type of molecule you see. Otherwise normally, as I said here, coupling to proton is usually not observed, except in symmetric molecules like that. Now, we saw boron NMR. Well, let us not worry about it. Let us see proton NMR. What type of proton NMR we get? It's a very interesting thing. I'm sorry. See what type of things we are going to see is in the proton NMR of this one. What we are seeing is one H NMR is coupled to boron. When you are observing boron, you are seeing coupling to H4. We got that 14641 intensity pattern. Now let us see, we analyze the spectrum. There are strong 4 peaks here. What are they? Why is it coming? What is this coming from? And there are also small peaks here of equal intensity. Fantastic spectrum, all peaks are of equal intensity here. If you carefully see, these 4 peaks I would say is because of B11. B11 as you know is a spin is 3 by 2. So 2Na plus 1 if you put, N is equal to 1, I is equal to 3 by 2. So how many peaks you expect? 2 will cancel. So 3 plus 1 only you get 4 lines. So for spin 3 by 2, 2 into Ni plus 1, N is equal to 1, 2 into the 3 by 2. So 2 cancels, you will get only 4 peaks. And these 4 peaks are for B11 and of equal intensity, and this separation adjacent line gives you 11B and proton coupling. What about B10? Its spin is 3. If you put 2Na plus 1, what happened? 2 into 6 plus 1, 7 peaks you should get. But remember, 7 peaks you should see here. 1, 2, 3, 4, 5, 6, 7. Fantastic. You are seeing all the 7 peaks of equal intensity. But what is the intensity compared to this and this? This is much larger than this. The reason is B11 is 80% abundant. Okay? B11 is 80% abundant and B10 is 20% abundant. As a consequence, the intensity ratio is 20:80. Okay, 2:4 intensity ratio should be there. And that is what you are seeing. And this gives you boron 10 proton coupling. This is what we observed when we observed the boron resonance, right? Same couplings we measured. We did 2 experiments we have to do, boron 11 and boron 10 experiment. On the other hand, in one proton NMR experiment, all the couplings of boron 10 to proton, boron 11 to proton can easily be seen here. Let us go to nitrogen 15 spectra. Something difficult to observe. You can see nitrogen 15 NMR here, and you can see coupling to other things. Generally, it is very weak. Nitrogen spectra of some amino phosphates is very, very weak because nitrogen is very low abundant. So you need to acquire more scans. And of course, the majority of these problems have been solved now because we have different types of highly sophisticated, very sensitive probes for detection of NMR signals, like cryoprobes and etc. Even nitrogen 15 in natural abundance detection is also not difficult. I just wanted to show you nitrogen 15 in natural abundance can also be seen, and its nitrogen to couple to other nuclei can also be measured here. Okay, and especially in this case, this is a fantastic example. There are 4 nitrogen attached to tin here. 4 nitrogens are attached. All 4 are equivalent. And then if we do tin and this for nitrogen 15 NMR with proton decouplings, 4 nitrogens are there. All are equivalent. So its sensitivity is much better. No problem. But you see, the 2 chota lines you see here. What are these chota lines? They are satellites, but not carbon-13 satellites like this. They are satellites of tin. Tin has 2 isotopes. Tin, of course, infinitesimal, there are 3. We'll ignore 115. Tin 117 and tin 119. There are 2 isotopes are there. Each of them can couple to nitrogen. When your nitrogen is itself is a dilute spin, very low abundant, 0.37. But when you are observing that, we will not worry about that. With respect to this, now this is a low abundant spin. This appear as a satellite. Look at this. And you are detecting nitrogen. 2 peaks are appearing here, which are satellites. One correspond to tin 119 nitrogen coupling. Other is tin 117 nitrogen coupling. So that is the important thing. So now by using the ratio of these peaks, one can find out the presence of percentage of different this thing isotopomers present there, because intensity is very precise here. If you measure very precisely, you can find out the percentage of 119 and percentage of 117 both, and they are now almost equal. Now we'll see 1H NMR spectrum of natural abundant urea. What is the urea? It is a simple molecule, CONH2 twice, I think. Look at the 1H spectrum of that. There is a very broad peak coming. What is that coming from? Urea contains NH2 group. Nitrogen has 2 isotopes, N14 and N15. N14 is a quadrupolar spin. Generally, it relaxes very fast and gives a very, very broad signal like this. This is because of N14. And you will not be able to see other couplings at all here. It is so broad. Whereas nitrogen 15 is low abundant, 0.37%, but still it is a spin-off nuclei, and that gives us to a doublet nitrogen 15 proton coupling. You can see this is a proton spectrum, NH proton here. This is also NH proton, but N15H proton. This is N14H proton. This majority of the peaks like carbon 12H and carbon 13H, majority of the peak, strong peak is coming because of 14N attached proton. This is 15N attached proton. Okay, this is. So as a consequence, very weak in intensity, and nevertheless, measuring the separation gives you proton, nitrogen 15 and proton J coupling. That information is easily obtainable. Now you can compare the N14 and N15 spectra also you can record of this urea. CONH twice is there. Now N14 spectrum, if you record, it is very broad. Look at the width. It starts from somewhere here. Let us say you take the mid here to here. You will take half height. Peak width is approximately 50 ppm. It is a large line width. N14 is really broad because of quadrupolar nature of the spin. It is N14 is spin 1. Whereas central peak is N15. N15 is spin half. It is a spin half nuclei. That is why you are getting a sharp peak. See, compare N14 spectra with N15 spectra. N14 is many orders of magnitude broader than N15 spectrum. This happens actually. Okay. So if I look for the proton spectrum of NH4Cl, again, you see N14 can couple to the NH4, H4 protons. So N14 can couple to that, and then N14 can couple to proton. It gives like to 3 lines of equal intensity, like similar to CDCl3 deuterium coupled to carbon, you know. Similarly, now N14 is coupled to proton, you get three lines of equal intensity. N15 couples to this, and you get a small chota peaks, or small peak of lower intensity. That is for 15N proton J coupling. So all these things are very easy. Now N14 spectrum of NH4Cl, it is something very interesting. N14 spectrum, if I look at it, it is coupled to proton, 4 protons. As I said earlier, you must get, according to Pascal triangle and 2Ni plus 1 rule, you get 5 lands, 1464 in intensity, very beautiful spectrum. Measure this separation, you get 14N proton J coupling. You understand? Similarly, we got this example earlier also. Mercury is another interesting nuclei, but it has 2 isotopes, 199 and 201. But most of the time, what people use is 199, because 199 is spin-off nuclei, and 201 is quadrupolar spin that normally is not studied. So the most favored nuclei for all of us, if you are studying Mercury, is Mercury 199. Okay. So now proton NMR spectrum of dimethyl mercury is taken, neat sample without dissolving anything. Mercury 199 spectrum, you see, you get a sharp, this is proton spectrum, you get a sharp proton peak because of methyl protons equivalent. But mercury is there, that is coupled here. And you see the mercury is very small abundance, it is appearing as a satellite here. This gives you mercury to proton coupling. Understand? This gives mercury to proton coupling as satellites. I'm just giving an example. Simple molecule, 2 methyls are equivalent. Proton spectrum gives you a single peak, and coupling to it is only mercury, which is small abundant. You get satellites. Now I can do satellite, instead of satellite observation, I can do HGNMR also. If I do proton NMR, mercury NMR with proton coupled, you are going to get 1, 2, 3, 4, 5, this many peaks. Why? It is, remember, it is coupled to 2 methyls. Each methyl is going to split this into a quartet, and quartet of one quartet split into each another quartet, another quartet, because of another Me2. As a consequence, you are going to get 1, 2, 3, 4, 5, 6, 7 septet, which you worked out earlier. Remember, this is what the pattern you are going to get. And the same mercury NMR, when you see that, you can see mercury 199. This is the proton couplings. Now, if you do the decoupling of proton, we can see carbon satellites now. Another interesting thing. Look at this one. Here, mercury NMR proton coupled. Here you decouple protons, no proton coupling. So in principle, it must be a singlet, but there is a CH3 group attached. They give us to carbon satellites. Carbon to mercury coupling is seen. See, so beautiful, simple molecule. You can see proton NMR, you can see carbon, you can see mercury. In the proton NMR or mercury NMR, you see carbon satellites. In the proton NMR, you can see mercury satellite. All possible things to measure information, you know. Very beautiful things we can do here. On the other hand, you can do carbon 13 NMR also. If I do carbon 13 NMR, what is going to happen? Again, proton decoupled, there is no splitting, you get mercury satellites. Completely we are decoupled proton. Carbon is only coupled to methyl now, mercury, because of this single peak for carbon, and these two are mercury satellites. So silicon NMR, of course, tetramethylsilane I showed you once, and you can get in the TMS, this is the proton signal, these two are carbon 13 satellites, and these two are proton satellites, they are silicon satellites. So on the other hand, I can see silicon NMR with proton decoupling, then I get carbon satellites. Very interesting, right? TMS, tetramethylsilane, I can see silicon NMR, also possible. In this case, proton NMR, I get silicon satellites and carbon satellites. If I see silicon NMR with proton decoupling, I see carbon satellites. Very interesting things. On the other hand, I can see proton coupled silicon. Here, how many peaks I should expect? There are 12 protons, all are equivalent, remember. Apply 2ni plus 1 rule. You should get 13 peaks. This is called tri-decuplet spectrum. Tri-decuplet means 13 peak spectrum. In principle, you should count here. There are 13 peaks. Here is 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12. Maybe 2 peaks are hidden somewhere here. Very weak in intensity. And according to Pascal triangle, this is the intensity pattern. Huge. You can work out. You understand? So, I am just quickly going through lots of nuclei. Many spectrum can be analyzed. Basically, what you need is a foundation for analysis of the proton spectrum and carbon spectrum, how they are coupled. And another thing is, and there are dilute spins coupled, like weak abundant spin. I told you it appears as satellite, and you should know how to analyze the satellite spectrum. The classical example of dimethyl mercury. In that you can see mercury spectrum with proton decoupling, you will get carbon 13 satellites. You can see carbon NMR with proton decoupling, get mercury satellites. You observe mercury NMR, you can get proton couplings. All possible things you can see. So this is the beauty of NMR when you want to do multi-nuclear techniques, multi-nuclear NMR. Varieties of possibilities you can think of, varieties of experiments you can do, and get a lot of information. So with this, I think I am going to stop multi-nuclear NMR. In the next class, we will start moving to different topics. So analysis part of NMR of proton, carbon, multi-nuclear, a very all detailed information is given. Now start going to more advanced topic from next.